Hi, I'm Marnie Halpern. I'm a professor and chair in the Molecular and Systems Biology Department at the Geisel School of Medicine at Dartmouth. And my lab is very excited to be hosting Talia Marc this summer. She's our first Choose Development trainee, and it's a pleasure to be welcoming her into the lab. And I hope she has a really fun, productive summer and learns a lot.
Hello, my name is Dr. Cagney Coomer, and I'm a postdoctoral associate at the Geisel School of Medicine at Dartmouth College in Dr. Marnie Halpern's lab, where we use the zebrafish to study neurodevelopment. And I am the mentor, the Choose Development mentor for Talia Marc.
Hello everyone, I'm Talia Marc, a Choose Development Fellow at Dartmouth College. And today I would like to show you guys around the lab and tell you a bit about my project. But first, I would like you guys to meet Dr. Clinton Cave from Middlebury College, whom I am collaborating with.
Dr. Cave, do you mind explaining to all of us what you've done so far for this project?
Not at all. The idea behind this project is a collaboration between myself. I'm an assistant professor of neuroscience at Middlebury College, and Dr. Marnie Halpern at Dartmouth. And the idea is to try to add a little species diversity to this investigation that looks at a signaling process involving a family of enzymes that I've been studying for quite some time. These are called Gde enzymes, and I did my PhD dissertation on them, and I'm continuing to study them in my lab at Middlebury College. Briefly, they represent a unique signaling pathway that can function at the cell surface to regulate GPI-anchored proteins. This is a class of proteins that are tethered to the cell surface. So these enzymes come along and they clip that tether, and that causes those proteins to be released into the extracellular space. And that can have a number of interesting signaling consequences. We're studying these contexts we're studying the signaling pathway in the context of neurodevelopment. I am an embryologist. I study the formation and development of the early neural tube, which is the precursor to the nervous system embryonically. And we're trying to figure out whether or not these GDE proteins have the same role in my systems as it does in zebrafish. So I'm kind of bringing the protein of interest, and Dr. Halpern is bringing the zebrafish model.
So just to specify, what model did you use for this project?
So I've investigated these GDE proteins in mouse, and I'm currently investigating them in the avian neural tube, so the developing chicken embryo.
As for my project, I'll be using CRISPR-Cas9 to make a no-mutant zebrafish for the gene GDPD4 to investigate its role in radial glia patterning cells for neurodevelopment. So this is the zebrafish room where we keep all the zebrafish for people to do experiments and breed more zebrafish. And these are the babies. I help feed them sometimes in the morning, the juveniles, and it's just a bunch. How we obtain fresh embryos for experiments is that we set up a cross between male and female zebrafish using the tanks and filters you see, and this is what a zebrafish cross looks like. A good way to differentiate female zebrafish from male zebrafish is that the female zebrafish carry eggs, so their bellies are much more round, and the male zebrafish are slim and have yellow bellies. Here are the different developmental stages of embryos. As you can see, they grow quite quickly over a 24-hour period. These are Casper Zebrafish, and they appear ghost-like because they have 2 mutations that eliminate pigment, so you can see right through them. I hope you enjoyed my tour of the lab and an intro to my project. I'm looking forward to learning more about zebrafish neurodevelopment, and thank you to the Society of Developmental Biology for giving me this lovely opportunity to being a research fellow. Goodbye!